Hi, everyone. Welcome to today's webinar. Textile Exchange is very, very happy to partner with the Organic Cotton Accelerator with OCA and Global Organic Textile Standard, GOTS, to present this webinar to you today. The topic that we're going to be talking about is looking at GMO screening protocols or a new screening protocol for cotton and textiles. Luckily, we have experts with us who will be presenting on the content for this. Just a couple of things that I want to mention ahead of the content today. We are really happy to offer this webinar. I also want to just acknowledge the role that Joy Saunders has played in kind of bringing this partnership together. She's really been the one in the background, pulling all the slides together, making sure that everybody knew what their roles and responsibilities were. So appreciate that work, Joy. A couple of housekeeping things I want to mention. We will, as always, Textile Exchange will be recording this webinar. We will be providing a link to the recording as well as a PDF of the slide deck, and you can expect that to be sent out tomorrow. So just look forward to that. We will also be holding a Q&A session at the end. So remember your questions through the course of the presentation. You've got a couple of different options to submit your questions to us. On the right side control panel, there's a question box where you can just type in your question and submit it. There's also a chat box that you can type in a question for us. We'll be monitoring those questions throughout the presentation, so feel free to just pop them in whenever they occur to you, and we'll go through them all at the end. To just give you a quick idea of what we're going to be looking at today, we have with us, the focus of the webinar today is really all of this work on GMO screening comes out of a bigger picture focus that Textile Exchange is looking at right now in terms of just improving integrity. And so we're going to be introducing what the recently delivered International Reference Protocol for GMO screening in cotton and textiles, what it means and when to use it. It's going to be a really technical explanation. You're going to kind of understand what we're looking at, what this means. But the real focus of this, I just want to keep everybody's mind on what we're really aiming to do is to improve integrity in this sector, improve the confidence in organic cotton, in organically grown cotton. And we're really excited to partner with OCA with this, as well as GOTS. Just speaking of OCA and GOTS, we have a couple of fantastic speakers today. We have Bart Vollard from OCA. He's the program director. We also have Rahul Bajakar with GOTS. He's the managing director. I'm Ashley Gill, the senior manager of industry integrity at Textile Exchange, and I'll be speaking just a little bit on our own standard. So without any further ado, I'm going to pass the controls over to Bart. And Bart, you can take it from here.
Thank you, Ashley. And hello, everyone. Welcome. Thank you to Textile Exchange for hosting this webinar and inviting us to share a little bit more about the International Workshop Agreement on GMO screening in cotton and textiles. I'm Bart Vollard, as Ashley just said. I work for the Organic Cotton Accelerator as the program director, and I'll share a little bit more about the reason why we, as the Organic Cotton Accelerator, initiated the development of this ISO protocol, the process we followed in developing it, some of the key findings, and also the future use of this protocol within our own programs, as well as how you could use the protocol in your supply chain or otherwise. Let me see. Oh, yeah, it works. So the reason why we started building this protocol was, and I think many of you are familiar with that, the fact that GM testing in cotton and textiles, in organic cotton and textiles, but also non-organic supply chains, has long been an issue of contention within the sector. There was a lot of questions as into how to exactly do this type of testing, from whether you should be doing qualitative testing or quantitative, and how exactly should that be done, and what type of products you could still send off to reliably test for GM presence. What about lint? What about dyed fabric and garment? And we wanted to provide more clarity and create a common language and practice on this topic for both the textile sector as well as the laboratories that perform these type of tests. So with the initial conceptualization support of GOTS, Raoul, thanks for that, and additional financial support of C&A Foundation, we decided to address this need and create this globally accepted method to serve the industry's interest in more integrity, also for consumers, and strengthen the sector. Of course, this applies to organic, but not only organic. There's also it's the entire textiles industry, and whoever wants to know whether GM is in their supply chain can use this protocol. This all started back in 2018, where we started with actually formally proposing to the International Organization for Standardization to develop such a protocol. And I'll share a little bit more about who we did that with. Was it the project lead? All the participants in the process in a minute. But before we do that, why did we choose this format? Why to go with the Organization for International Standardization? Well, that's because we wanted this protocol to be very credible and to be developed in an open multi-stakeholder approach. And we particularly opted for an international workshop agreement because it's a document that ISO produces through a series of workshops rather than through the technical committee process that others of you might be familiar with, which is going through your national delegations. It takes several years to develop an actual ISO standard, and an international workshop agreement is a way faster way to get to a consensus among market players and other stakeholders to a particular topic that you need an answer to relatively quickly. It also provides a nice starting point of a future standard if you'd want to. So it actually was a great way for us to, within a year time, get to a protocol with a big group of market players, still leaving the option open for a future standard if we'd want to. Now, the ISO was the way to go for many reasons, and one obviously is the credibility that ISO has as a international standardization body. Also, of course, giving a lot of visibility to this protocol. They set out the entire development process, which gives both transparency and ensuring that these key principles of standardization were applied. We did this with many different stakeholders, all of them from laboratories to cotton and textiles industry players, government agencies, standard bodies were invited and participated in the discussions and the actual workshop agreement. And then another very important point is that many laboratories are used to working with ISO standards and documents, so that highly increases the quality and the interoperability of the document that's produced. Well, once we started or initiated the ISO International Workshop Agreement development, we were looking for a strong project lead that could actually lead the technical work of and write the protocol, which needed to be a neutral laboratory with experience in the space. This was done by Wageningen University Research and their reference laboratory for GMOs in food and feed, also being a member of the European Network for GMO Laboratories. They did all the research, they did the analysis, they wrote the protocol, and they led really the development of this protocol that's available today. While the secretariat was done by the Netherlands Standardization Institute. NEN is the Dutch member body of ISO, So they met all the qualifications according to the ISO directives, and they brought in the necessary knowledge with regard to EU legislation, standardization, and accreditation. Now, in this entire process, we also had an independent chair, Sally Urban from Forum for the Future, and most important, over 80 participants from 23 countries participated, and really they represented, as I just said, all stakeholder groups. So we had the laboratories there, cotton and textiles industry, fashion brands, spinners, farm programs, academia, NGOs and foundations, all with a very clear process of commenting on the protocol as developed by Rikild. So, it's a little slow in responding to my mouse clicks. Anyway, the scope and objectives of this protocol. So I'm not sure how many of you have downloaded the protocol already. It's available on the ISO website, but essentially the elements it covers are four. First of all, the first objective of this protocol was to identify materials to be assessed based on the probability of actually and the possibility of obtaining good quality DNA from the materials. So that really means in what step of the cotton to garment and supply chain can we still extract DNA from cotton from sufficient quality to actually run a GMO screening. If you have not enough quality DNA available anymore, then it makes no sense to do this. So it was important to inform up until where in the supply chain you generally can extract that good quality DNA that enables this. Then it's important to also see what exact method for the DNA isolation would be recommended to get this DNA out of the products, out of your raw cotton, your fiber, yarn. And once that's done, to make sure that you're actually talking about cotton DNA. So what's a way you can actually control whether the DNA you've isolated comes from a cotton plant and not from soy or another origin. And last of all, how then do you get the most cost-efficient way of screening against all the known GMOs out there in the world to make sure that you really check all elements and GM events that are there. So you actually have a high level of assurance of knowing whether your cotton is free or not free of GM. Now, I want to stress the fact that the whole protocol discusses qualitative screening, essentially meaning giving you back answers like this sample, there is GM detected or there is not any GM detected. So in the scope of this protocol are all the steps that a laboratory takes the moment they receive a sample. So they prepare a sample, they isolate the DNA, they check the quality of the DNA and see whether it's truly coming from cotton. Then the GM screening starts, essentially giving you a yes/no answer, GM detected, yes or no, leading to the test report that you as a user will get. Out of scope of this protocol is, first of all, what happens outside of the laboratory, which is the sample collection. So it doesn't prescribe any measures as to the sample size to be, when you sample a bale of cotton or a farmer field. That's not in this protocol, but you have other standards and reference documents to be used for that. Another thing that's not covered is the actual identification, the quantification of GM events. So imagine in the event that you get a sample where GM is detected and you would like to know, okay, so what was the actual quantified the presence of this GM in the total sample, you'd have to do an additional test, and that's also not part of this current protocol. There's other material out there, like ISO 21570, that gives guidance on how to do this in a standardized way. Now, in this development process, Rekilt, of course, performed a few key activities that were also discussed with the participants. Then that led to several conclusions that really shaped the document. And I will highlight a few of the most important ones here, because it will give you an idea of what's in the protocol and some of the key findings that came out. First of all, of course, very important question that's long been debated in the textile supply chain is what's the most suitable DNA isolation technique with respect to being able to sample for GM presence as close to garment and dyed fabric as possible. I mean, preferably at garment and dyed fabric level. So what the results of Rekilt showed was that from seed up to unprocessed yarn and fabric, that means yarn and fabric that were not had not undergone wet processing, no bleaching, no dyeing, any finishing processing, none of that happening. So the grayish yarn and fabrics, there it was possible to actually isolate good quality DNA. However, it showed not to be possible to isolate amplifiable DNA in the yarns and fabrics, as well as the garment, of course, that was actually processed. So bleached, dyed, finished. And therefore, those type of products are excluded from the protocol. That's one key finding. Another one is the fact that it includes a actual suitable endogenous marker, as it's called. So actually a marker that will tell you whether the DNA that has been isolated from the sample truly comes from cotton. And this is a marker that works on all 4 cotton species that are commercially cultivated in the world. So it takes into account hirsutums, arboreums, herbaceans, and barbadans, and also all the hybrids out there. Then they were able to get to the most efficient set of elements or screening targets to be used to actually quickly control for all the known GMOs in cotton out there at a global scale, which is great because it allows for cost-efficient testing for all GMs in the product. And there's actually, so although quantification and identification is not part of the protocol, it also gives you some tools as to facilitate to identify what GM event you're actually likely to find in the sample in case you found GMOs there. And last of all, of course, the entire protocol has been tested on, again, real-life sampling, which showed that it confirmed the applicability of the entire procedure to all those stages of the supply chain up to grayish yarn and fabric. Now, of course, so this protocol is out there. It's published. It was in January that in an international workshop setting that the participants present that they agreed to a majority that to this protocol. So it has been published by ISO in April earlier this year. Everyone can download it from the website there. And as the Organic Cotton Accelerator, we use this protocol now in our farm engagement and development programs. So when we do testing for GM presence in the programs, we ask all our laboratories to follow this protocol and to report against it. Now, also, that same thing is something you can do being part of the Organic Cotton Accelerator or not. So if you want to use this protocol to test your cotton products for GM presence, it's very simple. You send the sample you want to test to a laboratory that is accredited against this specific standard of ISO, which gives you the assurance that the laboratory complies with general requirements for competence, impartiality, etc. So it's a decent lab you're choosing to do the analysis for you, a lab that knows how to work with ISO documentation. Then you explicitly have to request the use of this International Workshop Agreement 32. And all laboratories can access this, again, via the ISO website. So that should not be a problem. The whole protocol is written for labs to be easily understood and work with. It's a very technical protocol, so if you're not a laboratory, it might not be the most interesting document to read. And then, of course, we recommend testing for GM presence on cotton seed, on leaf, on raw cotton, the fiber, the lint, and the gray yarns and the fabrics, but not on these processed yarn, fabrics, and garment. Next slide. So that's how we are currently using it, and you can use it as well. But I do want to give some important last considerations when speaking about this protocol. First of all, again, we started this whole protocol development, first of all, because we want to enable stakeholders in the space to take reasonable precautions to avoid contaminations of their supply chain with GM materials. At the same time, I really want to stress, and I keep doing that over and over again, that organic is not a claim of absolute freedom from GMO contamination. It's the claim that any organic producer does not deliberately or knowingly use such technology. So it's really not meant to be, the protocol is not meant to be used in such a punitive way, and we do want to make sure that it is also not used in that form. Then there is the important consideration that technology and science are continuously developing, and we do not know what possible future techniques might bring. We know that quite some laboratories out there are working hard to improve the ways of isolating DNA from garment and dyed fabric. And yeah, we will follow those developments with great interest. We want to know about new claims in that space. But as a neutral platform, we really want to stress the importance of any such new emerging method that might be developed, or already developed by laboratories, to be validated externally by an independent organization and in line with a credible multi-stakeholder process. That's the whole reason why we chose to do this entire work with ISO. So again, this is let's say a state of the art for the moment, or as per January this year. These things can change, so there will for sure be updates to this protocol. And I also want to tell you that there's a lot of questions about it. We, as the Organic Cotton Accelerator, are there to help with any questions you have, but also to help accurately respond to interest on this protocol, because we really want to lead alignment on the communications on this matter. And that was it, I think, from my end. Thank you, and I'm handing over to Rahul and Ashley.
Thank you very much, Bart. Rahul, I will pass it on to you.
Wonderful, thank you. Well, to begin with, I have to thank OCA for taking up a fledgling idea that I had a couple of years ago. I was struggling as part of GOTS to find a proper protocol, and I had done some round-robin tests where results were not really the world's best. I couldn't come to any conclusion, realized we needed a much bigger study to be done and even perhaps develop a protocol. I wrote up a small project and talked to a few people, and I was extremely fortunate that the C&A Foundation and thereafter OCA took it up And thank you to OCA for supporting this fully, and even suggesting that it be an ISO IWA, which was not really my thought to begin with, but this worked out probably the best that it could be. So first, as I said, very happy that we have something. Like Bart said, it's not everything, but at least it's a beginning. So what does GOTS do now? Well, in this coming week, I will be sending off a communication to all our certification bodies, essentially saying that ISO IWA 32 will be the test protocol to be used for all GM testing. We will give a small implementation time, of course, because one doesn't expect this to happen immediately. Labs need to gear up and have an ISO accreditation. A small correction, perhaps, for Bart, as he mentioned, that only ISO 17025 accredited laboratories to be used. But what would be important here is to have this particular protocol as part of their accreditation scope. Because there'll be many laboratories with ISO accreditation, but we need to have labs with this scope within their accreditation. I will send this out to our CBs very shortly. In the GOTS revision process, which is currently on, changes will be made to our documents to say that this would be the protocol to be used and that it would be used up to unprocessed cotton stages. So that's immediately what GOTS is going to do. Like Bart mentioned, that there could obviously be future development in protocols and techniques, and we will think of what to do then. Like he said, it would be our endeavor to have a protocol that everyone can use, everyone has access to, and if there is validation, like he mentioned, with a new protocol, that would be welcomed, obviously. We are also mindful of the fact that this protocol should not be used as a punitive measure. We will, of course, in our communication with CBs, talk about what happens when something comes present. It is very clear to all the organic standards, including OCS and GOTS, of course, that GM is not expected, which means one does not expect any GM presence at all. At the same time, we are mindful that there could be inadvertent contamination, and we may have some discussions on what to do there. But the bottom line is no GM, full stop, in organic. So that's basically what I wanted to say in how we would be working with this ISO IWA 32. And happy to answer any questions either here or even later if you have any. So Ashley, back to you.
Great. Thank you very much, Rahul. So my statement that I wanted to make is very much in line with Rahul's statement. The organic content standard will also be adopting the protocol as the preferred testing method. So essentially what this slide is looking at right now is in the current version of the OCS, and I believe the language is similar in the current version of GOTS as well, is that testing for GMOs may be carried out and that it should be carried out at an early stage. But you'll notice that the actual testing method is not specified here. And what will be new coming with this memo that Rahul referred to, which we will be sending out a similar memo to certification bodies for the organic content standard, is that the IWA 32 is the required testing method if the risk determined by the CB warrants testing. No longer will there be allowed testing at the spinner, but only at the gen level. And this will be in effect after September 30th, 2019. So similar to GOTS, we'll be adopting this testing protocol as well. We are also in the process of revising the organic content standard. If you are on that list, you know that we have the International Working Group calls will be scheduled soon. We've actually adjusted the timing of the organic content standard revision to align more closely with GOTS so that we can address issues like this and make the standards continue to complement one another as much as we can. What we're going to be looking at with the revision is how to more clearly articulate the risk guidance in the certification procedures. So how the certification body assesses the risk in order to make the decision of whether to test or not, and as well as the sampling rate. And we're looking at having the standard release come out around March 2020, hopefully not too far off from the GOTS updated release as well. So that actually concludes my portion of the presentation as well. And we are now going to go into a Q&A session. And I think, Rita, you're going to be helping us out with that. Is that right?
Fantastic. That's correct. Thank you to all of our speakers for just giving a very informative presentation. A reminder to all attendees, you can add any questions in the question doc right now. And yeah, let's start. So I have a question that came in from Pedro. Is it still possible to collaborate on the development / improvement of the IWA?
Thanks, Pedro. So the IWA 32, as it's published today, it is what it is at this moment. So that development process has closed. However, there will be a review of the current international workshop agreement at least every 3 years. So that's certainly a moment where, again, new feedback can be included. But actually, anyone that is willing to update this agreement or thinks it needs to be updated can take the initiative to start that process again and make sure that an update happens faster if you want to. And if you have those ideas, questions, if you come to us, I'd be happy to hear from you.
Okay, fantastic. Another question that came in, the DNA that is extracted is quantified. Is there a minimum amount of nucleic acid to be able to perform the test?
So that's a great question. And again, I have to admit that I hired Rekild as a laboratory because they are the absolute technical experts on this front, not me. So coming from a relative layman in this field, I do know that when we assessed the quality and fit for purpose DNA to be extracted, of course, these type of factors were built in to assess whether we would have sufficient and fit for purpose quality DNA after the DNA isolation techniques, yes or no, if that helps. And if anyone else from the panel wants to step in and build on to this, please.
Yeah, I could add a few things. The protocol actually ensures that everyone follows the same method, and therefore, if at all there is, like I used to run a lab for 21 years, and I always said standardization is basically mistakes that everyone does at the same time, the same mistake. So it is a process. This protocol was found to give, if you follow this protocol, they found that this was the best way to isolate DNA. The stage at which they found the best DNA was up to an unprocessed cotton stage. So I, of course, would not know the exact amount of genetic material that was extracted. That if at all one needs to know, one might have to talk to Rikild. But suffice to say, the technique was properly investigated, and I think it's fairly robust for the purpose. That's why it was accepted as a protocol.
Great, thank you for that. We'll move over to the next question. Can you quickly explain the 4 DNA species of cotton?
Yes, absolutely. As in, with that we mean the 4 cotton species that are commercially cultivated across the globe. So obviously that's American upland or Gossypium hirsutum as one of them. That's the main chunk of cotton cultivated worldwide, but also Pima or Egyptian cotton, Gossypium barbadense. Then plus the 2 native Indian species, which is Gossypium herbaceum and Gossypium arboreum, which in India you also know as the Desi cotton. All of those 4 species were included in the testing, and the actual endogenous target that was identified in the protocol and is recommended in the protocol is one that will match or will show you presence of any of those 4 species and their hybrids, so any of the crosses.
Okay, great. Thank you, Bart. The next question we have, are there any plans discussed about the ability to test for newer GMOs like CRISPR, Talons, and ZFN's developed cotton?
That's a great question as well. So those were not included in the current review because to my knowledge, the GMO databases worldwide do not include those yet. But for sure, those things at the moment you'll have the test developed to control for that presence should be included or could be included. So that's something to keep an eye on.
Okay, great. Here's another question. What are certified companies to do if GMOs are detected? And what is the limit if GMOs are found?
I'll leave that to the standards on the call. So, Ashley, would you like to take those?
Well, yeah, I can start off. I can start off by saying that you should not find any GMO if it is a certified cotton to begin with. There is nothing called as a limit because it basically says should not be present. So I don't expect any GMO to be found present within a certified product. The thing is that the testing has to be done at the earliest possible opportunity. And in this case, it would obviously be at perhaps the gin level where the fiber where it's still in lint form. So at that stage, one can take a decision to accept or not accept. It's as simple as that. The reason why we were having problems is because of varying test protocols, methods. So there were labs using their own test methods. Some labs finding something present, others finding something absent. So therefore, a common protocol was a must. And that was why I was very keen on having something that everyone can use and basically take it as a reference protocol. Well, the answer is really there. You shouldn't have anything present to begin with. Ashley, if you have anything to add.
Sure. Yeah, I just want to mention that I think one of the points that you made, Rahul, is the one that I just want to emphasize, is that having a consistent methodology for the testing means that the information about how common it will be to find this type of thing, to what regions, if any, we are finding contamination. All of that information is going to be really helpful, definitely on the aggregate level, for both organizations like Textile Exchange, GOTS, and OCA to be able to start to build a picture based on consistent data. Because if we can't start with consistent data, then we're not really able to build that story of how prevalent this type of contamination is. And so this is really the first step in being able to understand really what, if anything is happening, where it's happening and why it's happening. And that's going to really enable us to help to build integrity and to build credibility even further. And so I think where we're at right now is looking more at the big picture, the value that this type of testing is going to provide. We're not necessarily looking at specific limits on certified companies. Not to say that that may not happen in the future, but right now that's not something that we've made any decisions on.
Yeah, actually, just to add again, sorry, but to add again, I also mentioned, having said what I just said, I said that using this protocol as a punitive measure is also something that we don't want to see. So what I will say is that the point is well taken. And unfortunately, not unfortunately, but because this protocol does not include any type of quantification, talking about a contamination or a limit is right now not right. And in any case, if a GM is found present, it has to be completely present. There is nothing called as a contaminated GMO. You cannot have 10 GMO in one fiber, 10 DNA in one fiber, of which one is GM. The whole thing has to be GM. So you understand that a limit part is something one has to talk about in bulk. Inadvertent contamination is something one has to think of. So there are many considerations. The point is being discussed, but the fact of the matter is that one should not find GM in certified organic, and that's the first aim. We will, of course, continue to discuss about what about contamination.
Thanks, Rahul. And Ashley, I really like also the answer you gave. It's very much in line with how the Organic Cotton Accelerator intends to use this protocol. It's very much a first step of this sector trying to align on how we measure things and to in order to improve integrity and to actually get comparable data out of the things we measure. And we, as in the Organic Cotton Accelerator, measuring the presence of GMOs in the farm programs we currently have in India, but also in the future in other parts of the world, will be using those data not for really to inform also likely upcoming regulations with regard to thresholds, because if we speak about inadvertent contamination, that obviously happens everywhere. And we know GM cotton is everywhere, especially in the countries where most of the organic cotton comes from, the US, China, India. And what does it mean to, what is that level or this quantified GM presence that we could still call inadvertent? What do we find on the best managed projects out there? We really want to make sure that whenever these type of rules or regulations are in the making, we as a sector can go to the table informed and with actual data, so decisions that are made will not hurt the sector too much, but at the same time will also allow us to keep striving for, obviously, I mean, the goal is being GM free, but how can we make sure that we don't take too rigorous steps? So it's the first step in organizing this sector further and having a similar way of measuring and reporting out.
Okay, great, thank you. We have a few more questions that came in. So as a personal suggestion, I believe a quantification and a definition of a set amount of DNA to be used in the test would be wise, because a PCR with 200 ng or with a 500 ng will produce different results when analyzing the values. So, I know that was more a comment.
I can take that. I think it's a good comment, and I think the protocol does specify a certain amount that has to be used. So it has taken into consideration these elements.
Okay, fantastic. The next one is for Rahul. Are you able at this time to comment any more on how it will be determined whether to test or not, and what the procedure will be if GMO is found?
Well, the protocol is a test method. So that is the method to be followed to determine whether any GM is present or not. And we will discuss with our certification bodies, and we have certain things that we will point out to them as to what needs to be done by CBs. And as I said, this advisory will go out to them possibly by end of this week or earliest possible. I think it should go out by end of this week.
And I just want to step in on this one as well, because one of the things that we're looking at whenever we draw up because I think that how the question of whether or not to test will be based on risk. And so essentially, and if you look at how certification works in general, anytime there's a sampling that's used, anytime there's selective type options like this that are given to the certification body as a way to kind of do further investigation, those are always based on risk. And so what tends to happen through the course of the stakeholder process is that we'll identify indicators of risk or factors of risk and provide those to the certification body as a way to determine the risk of potentially the input material that's coming from the farm or coming from the gin. I can't really comment right now on what those risk factors or risk indicators might be, but that's a process that we will go through with the multi-stakeholder group. The Organic Content Standard International Working Group will be making those decisions. We'll also obviously feed into the GOTS process to make sure that we're identifying similar risk indicators. But that's typically how we will determine both the whether or not to test and the sampling, how much testing actually happens. And then in terms of what the procedure will be if GMO is found, again, that's a decision that will be made based on what portion of the supply chain the testing happened, and really input from the users of the standards, from producers, to help figure out what kind of procedures and what kind of intervention makes sense in light of what we've learned. And we want to keep it out of the punitive stage and really try to keep that common goal of understanding and figuring out where this is happening and what's within the control of the farmer, the producer, the ginner or spinner, and what's not, and keep all of the any procedures that happen to be within the circle of what should happen based on how much responsibility there is. So all of those are decisions that will be made through that multi-stakeholder process that Textile uses to revise all of our standards. So hopefully that gives a little bit of a kind of a clearer picture of how those decisions will be made.
Great, thank you, Ashley. We had two questions come in that I'll combine together. One of it being, how common is it to find GMO cotton in certified cotton? For GMO as the result of drift, how does that impact things?
So I don't have an overview at the moment of how, so I cannot speak for, let's say, a global picture, but I do know it happens because the risk of contamination is everywhere, from seed production to in the farmer fields, to the mixing at the farmer stage, in ginning, in spinning. And so it's there's certainly contamination happening, and as we just explained, this protocol is a way of starting to understand it in a way that we, in a comparable way, so measuring apples, comparing apples to apples, and finding out where most of this presence starts happening. So we are actually able to work on preventative measures. Yeah, that would be my answer. Raoul and Ashley, any insights from your end?
No, that's what I would have said.
No,
Go ahead.
Yeah, I think you're absolutely correct. That's really where it is. We first need to find out a whole lot of information. There wasn't a protocol. Now we have a testing protocol that automatically starts to bring things together, hopefully. And yeah, we would know a little more than what we knew. And the way forward, I think, will come out of what we know or what we get to know after this.
Okay, great. We have a few more questions. Let's see. Considering that the method so far is considering the testing of raw products, seeds, fibers, etc., couldn't we quantify the presence of GM markers using a more large sampling method, i.e. testing 100 seeds and fibers and give a relative quantification?
Well, yes, but you're talking about a percentage of the number of seeds you have. That becomes, I mean, it's a percentage or a quantification, but what are we looking at? If you have a GM seed, it should not be part of the organic process. So if you find a set of GM seeds or a bag of GM seeds, you shouldn't be planting those along with non-GM seeds, is what I would say. So it's a little complicated. I wish there were easier answers, and I wish I could make a clear statement in terms of what is the limit and how do you quantify. The quantification part becomes very difficult because, to my mind, and as I see it, if something is genetically modified, it is genetically modified. You can't have a percentage of genetic modification. So when we talk about limits, we have to talk about what is a sample and what is a sample size. So if you're talking about, for example, 100 bales of cotton, and then if you have one of those bales as GM, then are you going to say that it's 1% and therefore either acceptable or not? I would say that that bale shouldn't be part of the organic process. Take the rest of the 99 bales. You know, that type of thing. So it's difficult to talk about percentages unless we know a whole lot of what is being found, where is it being found. And there are ways to tackle this. There are protocols which can be followed when you find something present. At the moment, I think it should just be that we now have a protocol to positively identify in a common way whether something has GM or not, and up to a stage of unprocessed cotton. We have to first obtain a whole lot of data over the next months, maybe a year, I don't know, to come to some other conclusions on other things such as contamination. This is my opinion at the moment as things stand now. Maybe Bart or Ashley can add to this if they have any other insight.
Yeah, I do. Thank you, Raoul. So two things. On the one hand, and I agree with Raoul, we will be, as OCA, for at least a portion of the programs we manage, start doing, besides the qualitative testing, also the quantification of once a sample is found to be positive for GM presence, quantify the actual that GM present in that sample, just to start gathering some numbers on the amount of GM present, whether it's 90% contaminated or 5 or 1%. And that's important because, of course, when you sample in most stages of the value chain, even at the seed level, because you will never test one single seed, you will always mix up material from multiple cotton balls and plants, and so it's always possible to find a balance between GM and non-GM. And now the reason why I wish it were also as simple to send off 10 samples, do a qualitative sampling, and imagine like 1 comes back as positive and 9 come back as negative. We could say, well, there's 10% GM presence, but that would be incorrect because this one sample found to be GM positive could actually have only a 0.9% GM presence in it. So we'd really, I mean, in order to speak about quantified presence, you would really have to do that that quantification followed but after finding a positive result from your qualitative screening. And there is actually protocols out there to follow. So ISO has some standard there. So it's already possible to follow again a standardized procedure if you want to go into quantification after the qualitative testing.
I think the only thing I would add is just to try to take the focus maybe back to the introduction that we gave in the beginning, is that the goal of this is to increase the integrity in and confidence in organic agriculture. And the goal of organic agriculture is not that farmers have 100% control over contamination on their farms, but that they are not intentionally using genetically modified materials, and that they're practicing agriculture in a way that follows along with the principles of organic. And so as we, as OCA and Textile Exchange, learn more about the presence of contamination or about the results of this testing, all of that information is going to be put into that bigger target or that bigger aim that we all share, which is to increase the amount of organic agriculture that's happening, to empower farmers to continue to move towards that. And so any use of this type of testing or this type of information that punishes those farmers for things that they're not doing intentionally, or that punishes gens or actors in the system, we want to be able to use that information to reach those bigger targets of increased integrity and increased competence in organic agriculture. And that's something that all of us are really keeping at the top of mind as we go through this. And so even though, there's definitely a lot of technical details to work out, and I'm not going to pretend that I even understand all of them, but just to kind of bring it back home that the purpose of all of this is greater integrity, and that we want to use as much information as we can to ensure that we're moving the industry in that direction.
Yeah, here, Ashley. That's perfectly said.
Fantastic. But there are quite a few other questions, and this webinar is ending at the top of the hour. So what I'm going to do is share all of these with our speakers, and they can email you directly. So everyone that asked one and didn't get their question and answer to, don't worry, we'll make sure that they are responded to. And yeah, at this time, I would like to take this time to thank our speakers, Ashley, Bart, and Rahul, and another thank you to Joy for helping to put this all together. This recording and presentation will be sent to all of our registered participants, so please be on the lookout for that email coming tomorrow, and you can re-listen to this presentation. So thank you again for all of your time, and if you asked a question, be on the lookout for a response coming in shortly from our speakers.
Thank you.
Thanks, everyone.
Thank you again, everyone. Have a great rest of your day and evening. Bye-bye.
Bye-bye.